Hello. And if you are here to see the advanced training presentation, Jellies, Man-O-Wars, and Dragons, some toxic marine life at our beaches, you're in the right place. Dr. Cindy Howard will be presenting this class, and she has been teaching at UHCL for 30 years. She teaches courses in ecology and toxicology, and she does research with students in Galveston Bay, salt marshes, and on the Amazon River in Brazil. She'll soon be leading 2 more of these Amazon tours, and one will consist almost exclusively of Master Naturalist passengers. Also, Cindy currently chairs the Environmental Science Department at UHCL. So with great enthusiasm, I now give you Cindy Howard presenting Jellies, Man-O-Wars, and Dragons. Isn't that a great title? Some toxic marine life at our beaches.
Thank you, Barbara. That's always very sweet. Whatever you come up with, thank you. I appreciate it. Okay, let's see if I can share this. Okay, so we were talking a little bit earlier. This whole presentation is inspired by Ann Anderson. We started talking about some Man-O-Wars and this new blue dragon thing over here on the right. That were starting to appear on Galveston beaches in the spring, and thought it might be a good idea to do an AT on it to just get everybody familiar with what these things are. So it's called Jellies, Man-O-Wars, and Dragons, some toxic marine life at our Galveston beaches. And the word "at" here, it's actually, all this stuff is living in the water along our beaches, and sometimes it washes up dead on our beaches. So "at" was probably the best word to use. So I'm going to talk about that and some not so toxic. That I want to also cover some not so toxic marine life is because we need to know the difference. We need to be able to tell the difference between what we need to avoid getting close to and what probably wouldn't be so dangerous. So let's go. The learning objectives are to figure out what a jellyfish is, which jellyfish can sting and why they sting, and some questions about why jellyfish even exist. Most people, and I know not you guys, but most people, if you had a presentation on jellyfish, they would have 2 questions. They would say, just tell me how to avoid getting stung, or how do I make the sting go away? And those actually could be a very short presentation. Because if you want to avoid being stung, just don't go to the beach, don't go in the water, and that's no fun. And if you want to make the sting go away, the present solution to that is to carefully remove the tentacles, whatever's stuck in you, with tweezers, and then put vinegar on it. It used to be that we were told to use meat tenderizer, and apparently that's fallen out of favor. So I don't know why, but vinegar is the preferred solution to jellyfish stings now. So in the next few minutes, we're going to explore what jellyfish, Man-O-Wars, and dragons are and how they compare with each other. They're all toxic. They all sting. How to identify the species that may be found along our coast, and especially along the beaches of Galveston. Which ones are toxic, which ones aren't. And then because I'm an ecologist and you guys are all interested in ecology, let's look at why they're important, why they're out there, what their ecological roles are in the marine environment. And you might be surprised at some of the answers. So let's dig in here. First, I want to start out with how they are classified in the animal kingdom. We're going to look at 3 phyla today. Phyla are the main body plan divisions in our animal kingdom. And a lot of times we're looking at vertebrates or crustaceans, arthropods, different groups. We got 3 totally different groups today that we don't normally talk about. The first 2 are phylum Ctenophore and phylum Cnidaria. The Ctenophore are the comb jellies, and the Cnidaria are the Coelenterates. They used to be combined into one phylum called Coelenterata, Silenterata or Silenterids mean cavity, just means cavity. And these were all put together, but they, I would say probably about 30 years ago, maybe a little longer, research on both of them found that the comb jellies really are very different from the Cnidarians. They have some similarities that we'll look at, but they're different enough to warrant their own phylum. And usually when we see taxonomists working on trying to divide or combine taxonomic groups, they're usually looking at the species or genus level. They're not usually looking at the phylum level. So this was a really big change, a big step forward. Both of these are what we call tissue level of organization. All they have is tissues. They have no organs. They have no systems. They're very simple. They're radially symmetrical. You get a little more advanced, and then animals are bilaterally symmetrical, which means you can draw one line down the middle, and then they're symmetrical on each side. But there's only one way you can do that. With the Ctenophores and the Cnidarians, you can draw that line anywhere in them, and you'll always have an equal half. So within the phylum Cnidaria, we're going to look at two classes, the class Scyphozoa and the class Hydrozoa, but you may also be familiar with Amphizoa. That's Anthozoa. That's the corals and the sea anemones. The Scyphozoans are true jellyfish, and the Hydrozoans include the communities of critters that we call blue buttons or Portuguese Man, and they're in different orders. So we're going to spend most of our time here, but when Ann and I were talking in April, there was a new toxic critter on the shores of the Texas coast, and that was a new sea slug or nudibranch called a blue dragon. So we'll take a look at those as well. So the ones we're going to focus on here are comb jellies, true jellyfish, blue buttons, Portuguese man-o'-wars, and blue dragon sea slugs. So let's start out with the ctenophores. What is a ctenophore? And this is an artwork by Ernst Haeckel, a German naturalist 100 years ago, who identified hundreds and hundreds of species. And a lot of the species that he identified were these little tiny things that live in the oceans. So these are quite fancy in terms of ctenophores, but you can see that all of them have these rows of cilia on them that we call comb rows. And so these are called comb jellies as a result. But you can see they have the chamber inside, just like all the Silenterids did. They have what's called mesoglia. And you'll see that the ctenophores and the Cnidarians both contain mesoglia. Mesoglia is two layers of cells that sort of like make the bread of a sandwich, and inside the sandwich is a jelly layer. That's where they get the jellyfish name. And the jelly layer can be very thin, as in the ctenophores, or it can be pretty thick, as in the jellyfish. They have an oral pole, that's the mouth. So we don't call it a mouth because they have no organs. And they have an aboral pole, which is the other end. There's only one opening. So for all the ctenophores and the Cnidarians, the food goes in and the excrement and the reproductive material comes out. And it goes in and out the same hole, okay, because it's only one opening. These are really effective predators on plankton. And they're the largest organism that uses cilia for locomotion, but they're still caught in currents, but they will use the cilia to go up in the water column, and then they'll sort of just float down in the water column. And as they float down, they open up this oral cavity or this mouth, and they just trap a lot of plankton on the way down. So they're pretty good in some places where you've got a lot of ctenophores. They actually can regulate plankton populations. Some studies have shown that. They don't sting, and this is important. So these go in the not so toxic group. We catch the two species that we have in Galveston Bay. We catch them frequently on our bay walks when we do seining at Galveston Island State Park. And we often catch a lot of these, and they can be handled. They look like snot. We tell the kids they're snot. They like that. But then when you put them in the water in the little half moons, you can see their little comb rows and how they move. And they're really beautiful, beautiful little organisms. They're pretty transparent. They're also bioluminescent. So if you disturb them in the darkness, you can actually get a little bit of a faint blue glow in the water. They're ecologically important, but they can be invasive. Apparently, the main species that we have here in the Gulf of Mexico has been accidentally imported into the Black Sea and has no predators there, and has really messed with zooplankton populations in the Black Sea. This may be the most interesting part of them, is that they have a day. It's called Ctenophore Day. And it's October 4th, because if you pronounce Ctenophore, some people pronounce it 1004, 1004. And so October 4th is officially Ctenophore or Ctenophore Day. That's the Ctenophores. Here are the two that live in Galveston. Both of them are pretty small. This one is about an inch, Barrow ovata, and Nematocysts is maybe an inch and a half. But we catch both of them in seines down there in Galveston. I know we catch them on the bay side. They might be on the beach side as well. The Nematocysts is a little more fragile and a little more translucent. The Barrow is a little thicker. This is Barrow with its mouth open. So you can see can open that oral pole up pretty wide. And then basically it sort of floats over its prey, and then it digests them. I'll go into how these critters digest stuff in a little while. All right, so let's move on to the Cnidarians. Cnidarians are called Cnidarians because they contain cnidocytes. And that's one of the big differences that separate out the Cnidarians from the Ctenophores. Cnidocytes are stinging cells. In fact, the word cnidocyte comes from the Greek for stinging cell. They have a mesoglia, just like their form is mesoglia, just like the Ctenophores, with an oral pore and an aboral pole and only one opening. Everything goes in and out of the same opening. They are radially symmetrical, like the Ctenophores are. But they have two body forms. They have a sessile body form that's a polyp, and they have a swimming body form that's a medusa. And here's a comparison between the two. So the polyp, they actually have exactly the same features, but in the polyp, this one's stationary and is is oriented with the tentacles facing up, and the medusa form is swimming and it's oriented with the tentacles facing down. So the mouth on the polyp form is here. The mouth on the on the medusa form is there, but it's got exactly the same features. The polyp is stationary or sessile. The medusa is swimming or floating. There are 5 classes in the Cnidaria. The Anthozoa are the corals and sea anemones, and these are polyps as adults, and they're all fixed in place. The Scyphozoa are the swimming jellyfish, and they are medusa as adults. Some of these swimming jellyfish go through a stage where they do have a polyp stage in their reproductive life cycle. The Staurozoa and the Cubozoa we really don't have here, so we're going to skip over here to the Hydrozoa. The Hydrozoa are colonies of both medusa and polyp forms, all in the same organism. So the stinging cells that are indicative of the phylum Cnidaria are depicted here. So here's a tentacle of a polyp, but they're also on the tentacles of the medusa as well. And if you magnify that polyp, you'll see that it's got a whole bunch of stinging cells in here. Each of these stinging cells is a cnidocyte. right there, cnidocyte. If you dissected out a cnidocyte, you could see that it has a little hair trigger type thing on it. This trigger is attached to an operculum, which closes up the cell under really great pressure in that cell. There inside the cell is what's called a nematocyst. Nematocyst is a structure that can be propelled out of the cnidocyte and then stick in a prey or anything that happens to rub up against these tentacles. And it has venom, and that the venom then can be delivered to the prey or the unlucky organism that rubbed up against it. So it's all tactile. So there's some organism that runs that touches this trigger, which caught, and then we'll move over here right now. So here's the operculum is closed, something touches the trigger, it causes the trigger to open up the operculum, and then this is like in a minuscule of a second, it releases the pressure, and when it releases the pressure, this thing sort of unfolds inside out, and it drives the stylet straight out of the cell with a bunch of force, considering this is just a little cell. And the stylet is attached to the string, and the string is attached to the part of the cell, the nematocyst. And this is full of poison, toxin, venom, that then gets delivered through the stylet into the prey. And you can see that happening here. So the stylet here is in the prey, and then the thread is either barbed or sticky, depending on the species, and the barb will actually stick to the prey, and which means that the prey cannot get that barb out of it. This is usually a toxin that will stun the prey, make it immovable. For us, it just hurts like crazy, but it can cause a prey organism to be paralyzed. So that's what they do. Jellyfish do this, corals can do it, sea anemones can do it, and the Portuguese man-o-wars, of course, also. They all have these nematocysts as part of their anatomy. All right, so let's take a look at the true jellyfish. Let's see how they're put together. So the true jellyfish are the medusa form. As adults, they are swimming. And so they are constructed of a bell. The bell in many of these species is, we call it muscular. These guys have no muscles. All they have are tissues, but they're able to pulse this bell so that it can give them some propulsion. Underneath the bell, we have the digestive cavity and the mouth. You can see there's not really the mouth probably is in there, but we have the cavity. And then the aboral end, there's no exit. Everything's going in and out through the mouth. So that's the bell. On some species, this is really sort of thick, and you can't really see through it. In other species, it's the jelly in there is pretty transparent, sorry, and you can see right through into its cavity. So there's the digestive cavity. Then it has tentacles. So you can see these are the tentacles. The tentacles are loaded with nematocysts up and down the tentacles. And then we have what are called oral arms, and these are also loaded with nematocysts. The oral arms also have cilia on them, so that any prey that is stunned by the nematocysts then can be directed to the mouth via the oral arms, the cilia on the oral arms, and then it comes into the mouth. So some of the jellyfish we have in Galveston, very interesting jellyfish. This first one is called a cabbage head. I'm sure you guys have seen cabbage head before. Most of the time when we see cabbage heads, they look like this. They're washed up on the beach. But if you ride the ferry over to Galveston, especially in the fall, you can look over the side of the ferry and you can see just hundreds of these guys just floating in the water. And they're actually pretty cool looking when they're floating. They're a little bit weird looking when they're washed up on the beach. So they are unusual when it comes to jellyfish because they have no tentacles. What you're looking at here are the oral arms right underneath the aboral end, the mouth. And there are some crabs that are commensal in here that sort of live on these oral arms. The oral arms do have nematocysts. That's how they stun their prey. But there are a lot of organisms that are really immune to their nematocysts. So you can find little young spider crabs and some young fish in there sometimes, and they will be not only eating on some of the plankton that the cabbage head has fed on, but some of these spider crabs will actually eat part of the bell. I guess the cabbage head doesn't mind it too much because it doesn't try to get rid of them. These feed on plankton mostly, and in the fall when there's a lot of red drum eggs in Galveston Bay, they're feeding on a lot of those as well. They are the favorite food of leatherback turtles out in the Gulf. Leatherback turtles are big. These guys are big, and they've got some biomass to them, and they are the favorite food of leatherback turtles. And I think you can see, we're always talking about plastic bags being confused. The turtles confuse plastic bottles, plastic bags, lots of plastic material. You can see this cabbage head jellyfish sort of looks like a big piece of plastic here. They are a delicacy food. They exist all over the world, and they are a delicacy food item in some Asian countries. What they do is they will collect them, dry them out, then process them in a brine, then dry them out again, remove the salt, cut them in strips, and serve them on salad. It seems like they'd be pretty tough to me, but they are enjoyed in some places. I don't think anybody's ever tried to eat them here. Some of the ones that are in the Gulf of Mexico, when you see them, they have brown spots around the edge here, but not all of them do. But if you see one with brown spots, that's not a different species. It's the same species, and it's not unhealthy. It's just some of them have some brown pigment. If anybody has questions at the end about how these reproduce, I will tell you, but it's pretty bizarre. So I'll save that to the end. Another common moon jelly around Galveston is, another common jellyfish around Galveston is the moon jelly, Aurelia aurita. And it's very easy to distinguish because you can see its four gonads sort of shaped in a horseshoe. Its bell is very transparent, so you can, looking through the top, you can see the oral arms below, you can see the tentacles through there. So not much to the biomass of these things at all. The bells can be up to 10 inches, 12 inches in diameter. Some have been found bigger, but the ones around here are sort of on the small end. They're not a very strong swimmer. They just basically float around. Sometimes you can have blooms. Blooms are like big swarms of these moonflower jellyfish. Moonflowers are found pretty much only in the wet moonflowers. I'm trying to go too fast, sorry. Moon jellies are found pretty much only in the Western Hemisphere. They're found mostly along the Atlantic and Gulf coasts, down around the northern part of South America, and a little bit on the Pacific coast of North America. They prefer temperate waters. They feed on plankton. Anything in the plankton. They're not very discriminatory about what they eat. And something really interesting about them is they tend to seek out water that has a really low dissolved oxygen concentration in it. The highest concentrations of moon jellies have been found in waters that have less than 2 milligrams per liter dissolved oxygen. That's really, really low. But they like that. They don't really like fresh water, but they have been found in very low salinity waters. And again, they're basically floating. And rather than swimming, And what they seem to be attracted to are these very poor water quality areas. They are prey of sea turtles and birds. Some fish eat them. And if you pick one up, it will sting you, but it's a mild sting. Again, if you don't mind having a mild sting, then knock yourself out picking them up. That's the moon jelly. The next one is the bay nettle. And this used to be called the sea nettle. It's the Chrysaora chesapeakii. And since 2017, this is what our bay nettle, or this is what our stinging nettle jellyfish is now. Prior to 2017, this one was combined with Chrysaora quincacera, which is what we used to call it. All of the books say Chrysaora quincacera, but there were enough differences between them that scientists decided to separate out these species based on some of these anatomical characteristics. So if you look at the picture, they really don't look the same. So they call this one the oceanic one. And then the bay nettle is the one that's in the estuaries in the nearshore Gulf. So it got Chesapeake's name because it was actually discovered, and the work that was done on it to separate it out was on individuals from Chesapeake Bay. The difference is the bell diameter is smaller. Ours is half the size of the sea nettle. It only has 3 tentacles per octant versus 5. So nobody's going to get close enough to count the tentacles per octant, but it has fewer tentacles and the tentacles are longer. It also has only 3 oral arms. Well, I guess they both only have 3 oral arms, but this one has long oral arms and the sea nettle has shorter oral arms. So, just taking a look at their morphology, they look different. And then it turns out they really are separate species. So this is a picture of ours, and you can see it's got very long oral arms and a very small bell on it. Most of the jellyfish stings that people get around Galveston are the work of this guy. And the Texas Parks and Wildlife, with their sampling of Galveston Bay and the nearshore Gulf over the last 40 years, have found that this is the most common jellyfish on our shores. And it's about 2 times more, it's found 2 times more often than any of the other jellyfish combined. So that's the one that, the stinging jellyfish that we most often encounter. The next one you may not have heard about before. This is called a pink meanie. I love the name. This is an interesting jellyfish. It can grow to its bell can be 3 feet in diameter, and its tentacles can be 70 feet long. So it's a wonder why we never knew it existed, because in 2000, just 20 years ago, there was a big moon jelly bloom off the coast of Alabama. And when they were investigating, moon jelly blooms happen not so infrequently. But when biologists were investigating this one, they saw this big pink jellyfish that was inside the floating around with the moon jellies. And they just discounted it at first because this often happens when a jellyfish from somewhere else gets picked up by an ocean current and it gets transported way, way far away from where it's supposed to be. And then, they eventually die out, and that's the last they're ever seen. But this one was doing some interesting stuff. It was actually feeding on the moon jellies. So the biologists collected a few individuals, and they contacted all of their colleagues in Europe to see if there was anything that looked like this over there. And apparently there was one on the coast of Turkey that looked a lot like this. So they brought some samples over there and they combined them. They did genetics, they did all kinds of stuff on it. And lo and behold, it was not the same species. So they decided this was a brand new species that they just had never seen in the Gulf before. This may be important. It was only seen that one time, but this may be important. It has never been seen in Texas, but it may be important because as they think this is more a tropical jellyfish, and as oceans get warmer, they're thinking we may start seeing this in Texas coastal waters. Why it's important is because it packs a mean sting, and it could disrupt the moon jellies have a role, and it could disrupt that. I love this picture because look at all those little fish that are staying away from the tentacles, but feeding on some of the plankton that the jellyfish is also feeding on. Okay, so nobody knows what eats it, if anything, at this point, because it just hasn't been seen enough or studied enough. But pink meanie. All right, so the next group, and it's really unusual for me to do a whole presentation and then ask questions. I'm used to getting questions as we go, but hopefully you're saving them up. So we're moving now from the Scyphozoa into the Hydrozoa. This is the last class of Cnidarians we're going to look at, and we have two orders with them that we find on our beaches. The first order is Chrondrophores, and the most common one of those that we see is called a blue button. When you see a blue button, it usually looks like this once it's washed up on the beach, but these are really pretty little organisms. They are not true jellyfish. They are hydrozoans, which means they are a colony. A true jellyfish is one single organism, but the hydrozoans are little colonies. So the colony for this one is very simple. It consists of a float, which is hard to see here because it's not like a Portuguese Man float. It doesn't stick up out of the water. It just sort of holds the blue button at the water's surface. And then it has numerous polyps, and the polyps then will make these tentacles that radiate around the float. And you can see this one looks like it has radial symmetry. You can really tell that from the way it's floating around. They're about 1 inch in diameter, very small, not big blue buttons. They're found in oceans all over the world. They're found pretty commonly in the Gulf of Mexico as well. Their tentacles contain nematocysts, but they are not very toxic to us unless you're very sensitive to them. You can usually pick these up. I'm not going to guarantee you won't get stung, but usually you can't feel it. They are also commensal with the fish species, and it seems like they are mostly food for sea slugs that turtles, I guess, leave them alone, maybe because they're so small. Those are blue buttons. And then the granddaddy that we talked about doing this whole presentation about is the Portuguese Man, because there are seasons where we see a lot of Portuguese Man individuals washed up on our beaches. Portuguese Man are Siphonophores, and the genus and species is Physalia because it's the only one in this whole group. It has a float that floats on the surface of the water and actually extends up maybe 6 or 8 inches, depending on the age and the species. So this is our Portuguese Man. There's a blue bottle related to it in the Pacific that's a little bit smaller than ours. So it got the name Portuguese Man because it's supposed to be reminiscent of these old sailing ships. I don't think the old sailing ships had all this stuff going on underwater though, but they do float around. They are at the mercy of the wind, the tides, and the currents. They cannot propel themselves at all. They're a colony of polyps and medusoids. So we've talked about this phylum having polyp adults like the sea anemones and the corals, medusoid adults like the true jellyfish, and then we have this group that have both of them. So in this drawing, we have the Nematocysts. That's one group of polyp. Well, that's actually one polyp. The original polyp makes the Nematocyst. And then we have underneath the Nematocyst is the digestive cavity. And we have gastrozoids, which are polyp, also polyp individuals, not organisms, individuals that are responsible for bringing food into the digestive cavity. Then we have gonozoids, that's this structure hanging down here. These are medusa structures, and these are the reproductive parts of the Portuguese man of war that I'll talk about on the next slide. And then we have the fourth kind of zoid is called a dactylozoid, and these are the stinging tentacles. And then I have to identify this guy. That's the unfortunate fish that's about to run into them. So this is a colony of organisms. It's not a colony of organisms. It's a colony of zoids that all originate from a single larval organism. So it's not like there are all these different individuals that have been put together in that colony. It's a colony of zoids all produced by the single larval form. So here's the life cycle. We have an adult. An adult Portuguese man of wars are either male or female. And that's easy to understand because they arise from a single cell. So a male and a female Portuguese man of war will produce sperm or eggs, and then those will mix in the water, although nobody has ever documented how the fertilization occurs. And then they form a fertilized egg, and then a larva develops from that. And the larva is just a medusa without any tentacles or anything. That larva then starts to differentiate, and very quickly, within hours, because it has to do that, it will start developing some of the other zoids. So the first zoids, well, one zoid starts with it. The first thing it starts to do is, I'm sorry that those words keep coming up. I don't know if they're coming up on your screen or not, but the first thing that this larva does is it starts making the float. And then right after and so you can see that happening here. And right after it starts making the float, then it makes a protozoid so it can start feeding. So the most important parts of this early larval stage are making the float so it can sort of hang out here at the surface, and then it needs a way to eat. And so as you can see, as it develops here, and this isn't a matter of days, all this happens. It's getting a little more complex. It's adding on gastrozoids, and then it's adding on the tentacles. And eventually, after a number of differentiations here, you can see it's got a well-developed float here. Now it can float at the surface, and then it just keeps creating zoids, and then you have an adult. Pretty interesting. So this one does not go through a stage where it is a sessile polyp. Some of the hydrozoans do, but this one does not. Some interesting facts about Portuguese man of war: that nematophore is full of carbon monoxide. It floats at the surface, but and it's okay to be out in the air, but the rest of the organism has to be in the water, has to be submerged all the time, or it will die. It cannot propel itself, it can only drift. So it's at the mercy of whatever weather events are going on, or currents it gets caught in. And this is why they can be basically transported all over the oceans. The sails can be left-handed or right-handed, and that's determined by, so the whole thing is a little bit off. These are radially symmetrical animals, but the construction of the zoids around the float will make it either lean to the left or lean to the right. And it's not really known how they decide whether they're going to go right or left or not. but the studies on them have shown that in a group, in a big flotilla of Portuguese man-o-wars, there'll be about half left-handed and half right-handed. And what that does is if the winds change direction and some of them get thrown up on shore, then that means the other half will stay out at sea because they will float in different directions because of that. But that's just hypothesizing. Nobody knows that for sure. The tentacles in some of the big Portuguese man-o-war have been measured at 165 feet long. When you think about that, if you're out swimming and you see a Portuguese man-o-war, 30 feet away, that doesn't mean you're out of danger for reaching its tentacles. The nematocysts, and this is true of all of the Cnidarians, but particularly true of the Portuguese man-o-war, because its tentacles are so long and they break off so frequently that that nematocysts in these tentacles are still active even if the tentacle is no longer attached to the colony. And if the Portuguese man-o-war washes up on the shore, it dies, the colony gets dried out, those nematocysts are still active. And all it takes is something to touch them, and it will cause the stylus to shoot out and bury itself into the skin of whatever touched it. These are related to the Pacific blue bottle. Ours aren't called blue bottles. They're technically different organisms, even though ours are also blue, but ours are a little bit bigger, have longer tentacles on them. And the last one is in a totally different phylum, but this also, we were talking about this in April because in April, the first blue dragon was found on a beach on the Texas coast. And they've been known around the world. They have worldwide distribution, but they've never been found on the Texas coast before. So that became a big news item. So I want to tell you a little bit about those. These are called Glaucus atlanticus. They're found not just in the Atlantic, but they're found all through the Atlantic, Pacific, and Indian Oceans. Until just recently, they have not been found in the Gulf, but that's about to change, it looks like. They float up and down in the water column. And the way they can do that is through their mouth, they will swallow a bubble of air, and the bubble of air will sort of reside in their stomach. And then they can let the air go, or as they need to position themselves in the water column. And they position themselves in the water column so that they can feed. They are what are called counter-shaded. Counter-shading is an adaptation that allows organisms that live in the ocean, and many fish and invertebrates do this, to be one color on their ventral side and a different color on their front side and a different color on their back or dorsal side, so that they can stay camouflaged. So this is actually the underside of the blue dragon. And so it's this really brilliant color of blue on its ventral or underside. And it's this real silvery type white color on its dorsal or backside. But most of the time it's going to be swimming upside down to reveal its blue side. So it's hidden from predators from above, and it is shielded from predators from below by its silver side. So it's sort of a wonder that it didn't develop those colors in the opposite way so that it could swim straight up. But it spends most of its life on its back, and it's very successful, so it probably doesn't matter. Most people think when they see these pictures that these guys are big, but they're not. And if you look on the internet, you'll see pictures you'll see this picture like on the left and a hand underneath it. And it's like the picture is 2 times bigger than the hand. Well, those are all Photoshopped. This is a very brave person with one on its hand. So they showed up here in, the first one showed up on a beach in April of this year, and it was picked up by person on the beach who thought it looked very interesting. He ended up, it was on a rock, and he thought it was a really cute little thing. So he picked it up and he put it back in the water. It did not sting him, but it could have. He could have had a very different memory of this little organism if he'd done that. So they don't sting unless they want to. So they are technically toxic. Some consider them venomous. Some consider them poisonous. It all depends on how you define these words. Okay, this is their favorite food. If you recognize this on the left, this is a Portuguese man-o-war. They like to feed on Portuguese man-o-wars. The blue dragon does not manufacture any toxins itself. Instead, when it's eating the tentacles of the Portuguese man-o-war, it's ingesting the nematocysts, and it stores them in its tissues and actually moves them, transports them to the ends of these fins, if you want to call them fins. They're not really fins, but to the ends of these appendages. And then if they are disturbed by something trying to eat them or threaten them, they can release the nematocysts, just like the man-o-war would have, by some kind of tactile stimulation, and the nematocyst, the healthy whole nematocyst, will enter the tissue of whatever was threatening them. And they're actually more toxic than the man-o-wars are, because they concentrate the nematocysts in these little appendages. They concentrate them out there. So when they give a sting, their sting can be 2, 3, 4 times more powerful than the sting of the Portuguese man-o-war. This is sort of how they do it. This is a drawing of the interior of a blue dragon. And you can see that it basically consists of 2 things: a digestive gland, which is all this white part. So all it does is eat. And then where it stores the nematocysts. So it's digestive gland and nematocyst storage. And of course it has a mouth. But other than that, it doesn't really have much at all. It's just a Portuguese man-o-war eating machine. But the important thing here is don't pick them up. They may be cute. They're not going to make a good pet. And they're not going to be cute if they sting you like a man-o-war would have stung you. So all of these critters make up a very interesting food web. The ctenophores that we talked about eat plankton. The bay nettles eat a lot of ctenophores. They also eat plankton, but their favorite food is ctenophores. So the ctenophores control the numbers and diversity of plankton. The bay nettles control the numbers of ctenophores and plankton. The blue buttons eat plankton. The moon jellies eat plankton and fish eggs. The pink meanies eat moon jellies. The Portuguese man-o-wars eat everything. And then there's the blue dragons that eat Portuguese man-o-wars, and they also like blue buttons. And so just to show you that this is true, I would like to see if this works. And somebody on the team, if you could please let me know if this is going to work. Can you see this video?
Yes.
Yes.
All right, it worked. Okay,
Beautiful.
so yeah, they are beautiful. Don't touch them. So here's one laying on its back. Can you see my cursor? Laying on its back, feeding
Yep.
on this. Actually, these guys are eating blue bottles, and this one's actually, it looks like it's showing you its underside, but this is actually its upside. And can you see he's got his head all the way into the nematophore of that Portuguese man-o-war. It looks like they've already eaten the tentacles off. This one's still working on the tentacles, but yeah. So they float around where the Portuguese man-o-wars are, and so the reason we may be seeing more of these, as well as more of the pink meanies, and maybe more of the Portuguese Man-o-Wars is that these are all sort of semi-tropical species. And as the Gulf warms, we're going to be seeing more of these critters surviving in the Gulf. So there we have it. Let me share my original screen again. There it is. Hope you enjoyed learning a little bit about jellies, man-o-wars, and dragons.
Wow, Cindy, that is so interesting. And if anybody is ever out in Monterey, California, the Monterey Bay Aquarium has a whole giant room with nothing but jellyfish tanks. It's magical. And your presentation reminded me of that.
You know, this picture on the left here is from an aquarium. You know, they put the right kind of lighting in and the backgrounds. And most of those aquariums, the jellyfish that's most commonly displayed in there is the sea nettles, the relative of the one we have here. So
wow. Well, we do have a few questions, and we're going to start with, and I don't know if it was Babette or Larry, and they had a couple of comment questions so if you want to unmute yourself, Brassfield, and ask your questions, go right ahead.
Okay, the first comment that I made is that I got stung by a Portuguese Man-o-War many years ago, and I didn't know what to put on it. And we went to one of the piers, and they had some hydrogen peroxide. They saturated some cotton balls and put it on there, and that caused the poison, I guess it was, that was injected in the skin to drain out, and that really relieved a lot of the pain. So hydrogen peroxide also, I think it was a stronger concentration than which you can buy at the drugstore. The other question, though, is have you heard of by the wind, critters that are called by the wind? No? Okay, never mind.
No, what is it?
I think it's some form of jelly also, okay, because they actually just go wherever the wind blows them, but it's not a Portuguese Man-o-War.
Yeah, you know, I know there's a lot more out there than what I covered here. I was trying to cover the ones we mostly would see in Galveston, but in doing that, I learned of two that, you know, we haven't seen in the past. So we may be seeing lots more in the future because most of these jellies are sort of tropical. So.
And that by the wind you can find in our Beachcomber guide. There's a little write-up on what the by the wind is.
Oh, it's in there. So
Yeah.
we already have them. Okay, sorry, I missed that one.
And
I'll
you'll
have to look
find
it up.
them every once in a while with the Portuguese Man-o-War.
I'll need to look them up. Thank you.
Okay, Jean Fisler, you're next.
No,
Dr.
they're
Howard.
not parthenogenic.
Well, I knew that. I looked it up. But I noticed at least 3 terms you introduced today. Start with the silent C, right?
Yeah.
Ctenophores and all that. And I understand the whole Greek naming system and going through the Latin, but I just wondered if there was any reason there were so many of them. Is it one guy in charge of all this stuff, or is it just a coincidence?
Well, I mean, you could go back to Linnaeus because he was the one that named these originally. And they, you know, everything was Greek and Latin to anybody educated back there. And so then I guess you'll have to go back to the Greek and Latin and ask them why they start everything with a C. I don't know.
I get that part. I just wondered if this guy had an affiliation with the silent C, or if there was just a coincidence. That's all. Interesting.
I don't
There's a
know.
lot of it. Okay.
You know, it all both of these still refer to chambers, to those open chambers. So it could be the CT is part of that.
Okay. Thank you. Good show.
Thank you.
Great. And Karen Hazen, if you would unmute yourself and ask your question.
Yes, you had mentioned that the cabbage heads are, that the leatherback turtles like to eat them. And so and you said they were large. So I was just wondering what the size typically are for those cabbage heads.
Yeah, I forgot. They can have a bell on them about 12 inches.
Okay, thank you.
And they weigh, if you pick one up, they can weigh, I don't know, 5-6 pounds. So yeah, they're pretty substantial.
Okay, next we have Suzanne Jurek. Hi, Cindy.
Hi,
When you were
Suzanne.
when you were talking about the Portuguese Man War and you were talking about their development from egg and how quickly that happens, what kind of lifespan do these guys have all total?
They can live a couple of years. They don't live forever, a couple of years.
Thank you.
Great. And Pam House, you had a couple of comments and questions.
Okay, hi Cindy. My first question was, so how did they differentiate between zooids that live in colonies or simply differentiated parts of a whole? I think Jean likened it to, yeah, how do you make that different from having arms, legs, and noses?
Well, okay, I think I'm understanding your question. They are, so you can have a functioning organism that is missing some of the zooids. You can have a functioning organism that only has a few zooids. As the colony grows, you're going to be increasing the numbers of these types because it's just growing. And
I think my question is more as how do you decide that it's a colony and not just parts of a whole? I mean, because you're describing a life cycle where, you know, it all comes from one thing and it differentiates into other things. How do you decide those are other things and not just parts of a whole?
that's a great question. And I think, you know, calling these colonies is probably makes it confusing. They call them colonies because each of these little each of these little units has its own function and it functions by itself. And you can pull that unit out and it will still function by itself, at least for a little while. You know, it can't feed itself,
Okay.
but like the tentacles, they can function on their own. Those are made up of all those dactylozoids, and they can function on their own, even if they're separated from the colony. They're not going to get any nutrition after a while, but I think that's it. That's why they call them a colony, but they're genetically identical. They start out from that one fertilized egg, and you know, it's male or it's female, and whatever its genetics are, it's going to have it's going to be cloned through these different zooids in the colony. When you look at a Portuguese man war versus a true jellyfish, the true jellyfish, you can't really pull parts out of it and they will continue to live.
Okay, great. That makes
I
great
think
sense.
that's probably it, but you're
Okay.
right. Calling it a colony, the way we think of colony is that there are lots of different organisms living together. And that's not the same. They probably do need a different word for it.
Okay, great. And then my second thing was a reminder that I really would like to hear more about the kinky sex of the cabbage jellies.
Okay, you want to hear it now?
Sure.
Okay, so one cabbage head, so they're hermaphroditic, but you still need two cabbage heads to mate. So one cabbage head will shoot the sperm out of its mouth, and that sperm will be caught by another cabbage head. It doesn't spread in the water, it catches the sperm. And then that cabbage head it'll mate with that cabbage head. And then within a couple of hours, the larvae will drop out of the second cabbage head and float down to the bottom, wherever the bottom is, and will attach to the bottom and become a polyp. And then it will last a few days like that, and then it will be going through a process of it's not like molting, but if we were to talk about crustaceans, maybe it's like that. So it goes through these changes, and then it's called strobilation. And at the end of that, then it releases from the bottom, and it becomes a medusa, and then it will mature into an adult. And it happens incredibly fast. I mean, this is all within a couple of weeks. You're going from basically sperm and ovum to a young cabbage head. I guess they have to because they're going to get eaten if they don't. So I think the kinky part is one of them shooting the sperm into the mouth of the other one. I think that's the kinky part. So
Great, thank you.
Okay, Ralph, your question?
Yes, thank you very much, Cindy. I appreciate your presentation here.
Thank you, Ralph.
You've talked about a little bit, but I guess you'd be interested to hear more with the, climate, warming oceans, increasing pH, CO2 levels. What changes do we see, arising in the Gulf of Mexico with these species?
Well, I think what they're expecting is that, these guys float. Some of the jellyfish can actually swim, but most of these are floaters, and they end up living where the water is most conducive for them. So, if they like warm water, they're going to be attracted to warm water. If they like low pH water, they're going to be attracted to that, and they're just going to basically start floating in those directions. And since most of these the nastiest jellyfish out there are in the cubozoas, and we don't have those. Those are in tropical waters, mostly in Southeast Asia. But there's some fear that, if all the oceans are warming and these cubozoans get caught in those ocean currents, they could end up in the Gulf of Mexico too, because Gulf of Mexico gets those ocean currents. So it's basically they're floating in currents that are conducive to them. And if we get warmer, we're going to get some of those organisms. Did I answer your question?
Thank you very much.
And Beverly had a question. Beverly, if you want to unmute yourself and ask your question.
Okay, actually, it's Ron. We share.
Hi, Ron.
But,
Well, I'm sorry, Ron. Ron will unmute himself and ask his question.
but I look like Beverly. Anyway, my question is that when we moved in to our house on a canal, every summer we would have cabbage heads. So they'd swim around and bump into the docks and stuff. Then one day, one summer, after we'd been here about 10 years, we had a bloom only in our canal, and it looked like the canal had froze over. Texas Parks and Wildlife came out, the papers came out, it was in the papers. The next day, they were all gone. Since then, we've had very few cabbage heads in the canals where I live. So it was crazy.
They ruined the neighborhood.
Yeah, I know it. Any thoughts?
No idea.
You could almost, it looked like you could walk across the canal on these things.
Wow.
Not that many.
Well, I know that the cabbage heads are more temperate than the rest of these are. And I'm just surmising here. I'm not a jellyfish expert, but if we're seeing more tropical species come in, we may end up losing some of the more temperate desiring species, and the cabbage heads are more temperate desiring. So
The other next question is that I had a 75-gallon tank that I put fish I would catch in a canal into it. And I had some fish, triple tail, some moonfish, and piggies, of course. And I put about an 1.5 inch, 1.75 inch diameter cabbage head in there just for looks. And within 30 minutes, every fish in that aquarium was dead. And
wow.
within the statement that, cabbage heads don't have tentacles. How could he have done that?
They have nematocysts. The nematocysts are in the oral arms. So if the fish bumped up against the oral arms, they would get the nematocysts.
Yeah, okay. Enough to kill? I mean,
Well,
any fish?
it can, because well, it depends on what kind of fish and how big they are. But they don't hurt us mostly. That apparently, if you get them on your hands and you get them in your eyes, they're a real eye irritant. And there's some people that react to the nematocysts on their hands, but for most of us, they don't bother us at all. But they can affect fish.
Mm. Now it's Beverly's turn.
And I want to interject something. A couple of weeks ago on Turtle Patrol, we counted over 300 man of war on the beach.
Wow, that's a lot.
We're over on Surfside, and some of them were very small. So I don't know if the you know, I don't know what happened, but there was a bunch of them. That was just an FYI.
Yeah, no, well, you know, they're at the mercy of the winds. If we have strong southeast winds, that'll bring them in.
Yeah.
The ones with the sail that are going that way, the ones with
Yeah.
the sail going the other way are going to head off to Florida. So
Yeah,
Yeah.
it was pretty cool. I mean, we were riding, we weren't walking, thank goodness.
I guess they're pretty good at tacking though.
Okay, Jean has another question.
And then I'll stop. But yeah, one of the things that 2 weeks ago, we had some tremendous onshore winds. Is that the right term? And the beaches were covered with all kinds of ocean going stuff for about 5 or 6 days. But my question, Dr. Howard, is you mentioned one of these organisms goes up and is able to gulp air, and then it takes that air and uses it to find its level in the water column, but you know, the desirable level.
Mhm.
So here's the little chicken egg question. How in the heck did it get up there to get the air to begin with if it didn't have the air?
well, so that's the blue dragons that do that, and they can
Yeah.
swim. These are nudibranchs.
Oh,
They
okay,
can swim.
okay. He's okay. Then he uses the air to make it easier on
Yeah,
himself.
yeah.
Okay.
they can swim.
See? Thank you.
You're welcome.
Thank you so much, Cindy. This was another wonderful presentation, and I hope you have been watching the comments because everybody is agreeing with what I just said. And then the other thing I want to tell you is that on this wonderful warm afternoon, we had 68 people attending the presentation, which is pretty awesome. And so thank you. Now I'm going to turn it over to Ellen, and everybody can be done with today's presentation. Thank you, Cindy.
Thanks for letting me do it. I appreciate it.